Dear colleagues, uh, let me take this opportunity just to proceed. Sorry if I have bothered you again. Development of autoimmune inflammatory diseases brief scope of primary factors extending on molecular mimicry. It's about 87%. Is a consequence of lack of self-tolerance, which can arise due to defective function of any of the features contributing to the unresponsive status of immune system toward self. In American style and auto in British style antigens. It's the same. Auto. So autoimmune responses resemble normal immune responses to pathogens as specifically activated by self-antigens or autoantigens and give rise to autoreactive effector cells and to autoantibodies against the self-antigens. When reactions to self-tissues do occur and are then improperly regulated, they cause a variety of chronic syndromes called autoimmune disorders or disease, it doesn't matter. Meanwhile, myocarditis can be defined as the inflammatory process affecting the muscular tissues of the myocardium or just the heart. And then is being transformed in a stepwise manner into dilated cardiac myopathy. Myocarditis is considered an important cause of sudden death, as has been shown by autopsy-based studies in the general populations. Regardless to its etiology, the acute inflammation may progress to subacute and chronic stages, and finally to tissue remodeling, fibrosis, and loss of myocardium physiological architecture and contractile function finally. The latter, in a process of chronic damage, corresponds to development of dilated cardiac myopathy, DCM. The gold standard today for final confirmation of causative reasons for AIM is the cardiomyocardial biopsy analysis. CPM in turn comprises the genetic information together with the phenotypic and environmental factors to yield the tailored healthcare for each individual and remove the limitation of the one-size-fits-all therapy approach, so-called canonical standard due to Hippocrates therapy approach. This provides the opportunity to translate therapies from bench to bedside to diagnose and to predict disease and to approved to improve patient-tailored treatments based on the unique signatures of a patient's disease. I mean, signatures mean molecular profiling. New targeted therapies for autoimmune and inflammatory diseases would require greater understanding of a patient or a pre-illness person at risk to get the therapy personalized for either of those subsets, more precisely for specific biomarkers as the target. Look, for today, no idea about the tomorrow. It's types, I mean general, global types of biomarkers for today. The most influential and valuable, for sure, diagnostic, monitoring, predictive, and susceptibility risk. Biomarkers remains the holy grail of platforms, algorithms, and protocols, which are the crucial for PPM. Meanwhile, the utility of predictive, prognostic biomarkers and other biomarkers of the next step generation to monitor and to manage chronic autoimmune inflammation is dependent of lots of parameters, but on 3 strategic parameters, which must be carefully assessed: sensitivity, specificity, positive predictive, and prognosticable value. So, accurate, precise prediction is crucially vital for prevention of autoaggression. And the targeted treatment could thus be given to those individuals who are most likely to develop the disease. In terms of personalized precision cardiology, it's a pre-illness persons at risk. AIM is just one of the chronic organ-specific or tissue-specific autoimmune diseases resulting in the destruction of cardiac tissues by different tools, including highly aggressive and destructive autoantibodies. The crucial role in the development of cardiac-driven autoimmunity in AIM is cardiac myosin, one of the most important self-targets in AIM. The latest concepts demonstrate that anti-CM cardiac myosin autoantibodies play a significant role in the development of cardiac autoimmunity conditions. I have already told you in the previous. Several groups of basic researchers, translational researchers, and clinical researchers have shown that a subset of patients with AIM and of their symptom-free relatives has circulating heart-reactive autoantibodies. Those antibodies are directed against multiple antigens, some of which are strictly expressed in the heart, in the myocardium. For example, organ-specific for the heart, Others are expressed in heart and several skeletal muscle specific. Antibodies of IgG class, which are shown to be cardiac and disease specific for AIM, can be used as reliable markers of autoimmune pathogenesis for identifying patients in whom immunosuppression and or immunomodulation therapy may be beneficial and their relatives at risk. Moreover, some of those antibodies may also have a functional role in patients, as suggested by in vitro data as well as by preliminary clinical observations, though further work is in progress to clarify this much important issue. I have already mentioned it in the previous lecture, but I was restricted in time. Antibodies possessing with catalytic, in this term, I mean in term of autoimmune myocarditis. Catalytic, I mean proteolytic activity. Possess to antibodies with a feature of functionality. Antibody linked proteolytic activity. About examples. The examples of antibody proteases mean in this term proteolytic antibodies, are IgG found in a series of autoimmune disorders to cleave targeted autoantigens and thus to act as informative biomarkers to monitor both clinical, canonical patients and subclinical, most the relatives, causes of the disorder. I have already mentioned about it, bronchial asthma, we'll just miss it. Antibody proteases in autoimmune myocarditis. In the overall population, 92% of AIM patients demonstrate the presence of antibody proteases. The activity of them markedly differs between AIM patients and healthy controls. Of greater interest is the evolution of antibody-associated targeted proteolytic activity at different stages of the AIM progression. For those purposes, we have been monitoring AIM patients, their direct relatives, and healthy controls, voluntaries, for around 2-3 years to have them tested for antibody proteases. Look, 70 and 20% of the AIM patients and their relatives were initially seropositive for canonical anti-cardiomyosin autoantibodies with no proteolytic activity, so-called disarmed or canonical antibodies. And less than 2% of the AIM-related relatives were initially seropositive for antibody proteases, but very low active. 75% of AIM relatives being initially seropositive for canonical anti-cardiac myosin autoantibodies and low active antibody proteases, and monitored for 2-3 years, have been demonstrating stable growth of the antibody-associated proteolytic activity during the time under the study. Being past subclinical and clinical stages. The activity of antibody proteases is first registered at the very early subclinical, no clinical, neither instrumental manifestations of AIM, status of IM. When antibody proteases are low active, the inflammation is practically minimized, zero, and the manifestation and thus moderate or absent. Initially, half a year, 2 years, 3 years. Healthy controls, AIM patients with clinical full-scale clinical manifestations. And it's broadly known, canonical, the antibodies play neither canonical, predictive nor discriminative role to affect the subclinical stage of AIM. In contrast, antibody proteases with the targeted functionality have proved to be greatly informative and thus valuable biomarkers to monitor AIM at both subclinical and clinical stages. Therefore, the proposed predictive value of CM, cardiac myosin, targeted antibody proteases for the development of AIM is being challenged. The translational, the design-driven translational potential of this knowledge is in the rational design of new diagnostic tools and new targeted therapeutics based on principles of artificial biocatalysts and biodesign. In this sense, targeted antibody proteases can be programmed and or reprogrammed to suit the requests and standards of cardiac muscle regeneration. Targeted antibody-mediated proteolysis is just an example illustration of the latter. Could be also applied to isolate, from Ig molecules, catalytic domains directed against cardiac autoepitopes or domains containing segments to exert targeted proteolytic activity. Antibody therapeutics have entered the center stage of drug discovery and development as a result of a major shift in focus of lots of biopharma companies. So, antibody protease design-driven bioengineering would offer the ability to enhance or alter the sequence-specific activity, of antibody proteases, to expand the clinical utility of the absolutely new translational tools. Of tremendous value in this sense are antibody proteases directly affecting the physiological remodeling of tissues with multi-level architectonics. For instance, cardiac muscle or myelin. while securing the request and standards of regeneration. So further studies on antibody-mediated cardiac myosin degradation and other targeted antibody-mediated proteolysis may provide biomarkers of principally new generation or even new biomarkers families. Thank you so much. It's just the prolongation of the first presentation. Sorry for having bothered you.